Hello students, today's topic is mapping of genome by use of DNA markers. In our previous session, I discussed about mapping of genes using linkage map. It should be noted that the genes used for creating linkage map were phenotypically manifested or observed. Example, eye color, body color in Drosophila, biochemical traits in bacteria. However, in case of complex organisms with large genome, the genes are distantly placed. Example, in humans, plants like maize. The gene sequences are interspersed with sequences of introns, repeating sequences, etc. Mapping of genes at these regions are difficult due to absence of markers. For a high-resolution genome mapping, additional genetic markers are required. DNA markers can be used as markers in such instances. The second main disadvantage of classical markers is that not all genes show heterozygosity or allelic forms. In such cases also, DNA markers can be used. DNA markers. DNA markers or molecular markers are a stretch of specific DNA sequence located at a specific site on a chromosome. These serve as landmarks on the chromosome to provide researchers with information of the proximity of the gene of interest. These markers segregate in a Mendelian fashion. For construction of linkage map using DNA markers, the basic principle utilized is linkage disequilibrium. Several markers are utilized and screened across the entire genome for a high-resolution genome map. It should be remembered that these DNA markers are not genes. DNA markers are used to construct linkage maps. The principle is same as discussed in earlier session. Linkage disequilibrium. Markers closely linked to gene are inherited together. The recombinant frequency is calculated and the markers are assigned to linkage groups using the LOD score. Procedure for constructing linkage map using DNA markers. First, collection of blood and isolation of DNA samples from members of the family. It includes both normal individuals and with individuals with certain disease or characteristics. The DNA profile is then constructed and scored using different DNA markers. The polymorphic bands are scored for all the individuals and correlated with the phenotype of normal individuals and diseased individuals. Markers that are in near proximity to the gene for the disease would be linked together and inherited together. Recombinants are analyzed and recombinant frequency is calculated between the DNA marker and the gene for the disease. Large number of markers are used and it is likely that one among them would be in close proximity to the trait associated for the disorder or disease. There are different types of DNA markers like RFLP, AFLP, RAPD, CAPS, SSRs, SSCPs, HAs, SNPs, ESTs, STS. They vary based upon the principle utilized. Some are restriction based, some are PCR and non-PCR based. However, for genome mapping, the markers must have at least two allelic forms. Based on this requirement, for genome mapping, we use RFLPs, simple sequence length polymorphism, SSLPs, which includes both micro and mini satellites, and SNPs. Restriction fragment length polymorphism, or RFLP. Restriction enzymes are a special class of endonucleases which cleaves DNA at specific sites. Each restriction endonuclease site has a specific sequence of target nucleotides. These specific restriction endonuclease sites are distributed throughout the genome. The length of cleavage size in certain restriction endonucleases vary among individuals. This is mostly due to point mutations. Hence, the DNA fragment length varies when such genome sequences are cleaved by restriction enzymes and the gel profile is analyzed. Example, in the figure on the extreme bottom, the first allele has three restriction sites for a particular restriction endonuclease. Hence, it gives four fragments of varying lengths. The second strand, allele 2, has only two sites for the restriction enzyme and hence gives only three fragments. This can be analyzed by means of a gel profile. The main disadvantage of directly scoring restriction enzyme digested DNA on agarose gel is there are many restriction sites for a particular restriction enzyme and hence would give many fragments of varying lengths. Out of these, only few are polymorphic. Example, in case of restriction enzyme EcoR1, the restriction site is repeated approximately every 4096 bare pairs apart. So, roughly on calculation, in case of human genome, around 8 lakh bands would be produced. In such cases, the scoring with agarose gel becomes difficult. Hence, alternative methods of scoring are used. The two main methods used for scoring an RFLP is utilizing southern blot hybridization technique and PCR typed RFLP method. In southern blot hybridization technique, the bands of DNA fragment on the gel are transferred to nylon membrane. These are then hybridized with labeled probes designed for specific polymorphic restriction endonuclease sites. These are then visualized and scored. In case of PCR-based detection, specific primers are designed for the polymorphic restriction sites and amplified by PCR. This is then digested with the specific restriction enzyme and then scored in age. The figure on your right is an illustration of these two methods. Analysis and interpretation of RFLP gel profile. As an example, we would take a hypothetical pedigree for inheritance of sickle cell anemia in a family. Sickle cell anemia is a recessive autosomal linked disease. The pedigree here shows the parents as carriers or heterozygotes. Among the progeny, one is a carrier. Since RFLP is a codominant marker, we can distinguish between homozygotes and heterozygote genotypes. Now, here the restriction site is located near this particular gene. Homozygous dominant have restriction site for the particular enzyme at one position. Hence, on cleavage, it gives two bands. The recessive genotype does not have a restriction site for the enzyme. Hence, it gives only one band. The heterozygotes give three bands, two for capital A allele and one for small a allele. So, on analysis of the marker profile in figure B, the genotype pattern for specific genotypes can be estimated by the unique pattern of bands. How is RFLP used in genome mapping? Here, first select a restriction site with two morphs and design a probe for this site. Example, in this case, probe B. After restriction digestion and southern hybridization, the gel profile is given below. Since morph1 has two restriction sites for the given restriction enzyme, two bands are formed which can be visualized by using the specific probe designed. In case of morph2, the restriction site for the probe designed is missing and hence only a single band is visualized. In case of a heterozygote, we would observe three bands. Next, we need to determine the linkage between the RFLP marker and gene D, which is supposed to be responsible for particular disease trait. A pedigree for the family is constructed and analyzed. Here, the individuals that are shaded in brown are affected individuals and the one in purple are unaffected. Blood samples are collected from all the living individuals in the family pedigree and analyzed for the RFLP profile. The gel below shows the RFLP profile of each individual in the pedigree. The first well shows the profile of mother. Here, three bands are observed. It shows the presence of DNA segment for morph1 and morph2. Hence, it can be concluded that the mother is heterozygote for this particular loci. Second well shows the profile of the father. Here, only one band is seen. It shows that the alleles at both the loci are for morph 2. Similarly, the genotype of all the progeny can be interpreted based upon the RFLP pattern, and it can be concluded that the ones which have three bands are all heterozygotes for the particular gene, and the one with single band are homozygote recessive. Now, next we relate the RFLP pattern with the pedigree as to whether the person is affected or unaffected. When we correlate the pedigree and the gene profile or the RFLP profile, we find that all the heterozygotes are affected individuals except the individual 8. It has a single band. That means, as per the profile interpretation, the individual 8 should be homozygous. However, the pedigree shows the person as infected. With this, it can be interpreted that the eighth progeny is a recombinant. Hence, it can be concluded that this particular restriction site is close to the gene of interest or locus D. Here, it can be inferred that the recombinant progeny 8 is formed due to crossing over that happened between the sister chromatids in the mother. The figure here shows an example of a RFLP marker DDE1 for site associated with sickle cell anemia. A single base pair mutation leads to loss of a DDE1 site in beta hemoglobin gene. The RFLP profile of the individual show that a normal homozygote would have two DNA fragments, one of 175 base pair and second of 201 base pair, whereas a person with sickle cell anemia, that would be a homozygote, would have only a single DNA fragment of 376 base pair. The heterozygotes would show three bands, one of 175 base pair length, second 201 base pair, and the third of 376 base pair length. The RAPD products can then be cloned and sequenced and converted into other type of markers like SNP and SCAR. The advantages of using RFLP as marker in mapping is its high reproducibility, shows codominance, and even neutral DNA variations can be detected and used as markers. The main disadvantage of this method is it requires high quality and quantity of DNA and shows less polymorphism. Simple sequence length polymorphism or SSLP. These are a series of repeated sequences of varying lengths that occur in the chromosome. Based upon the number of repeated units, they are classified in two types: variable number tandem repeat and dinucleotide repeats, are also known as SSR or STR. Both are tandem repeats and show polymorphism and are multi-allelic. VNTRs are variable tandem repeats distributed throughout the genome. The region in the genome that contains variable number of repeat sequences, generally 15 to 400 base pair, are called mini-satellites. These repeats would be around 20 to 50 times. Since they are multi-allelic in nature, they can be used as markers for mapping. The figure on the right shows a VNTR region in a small location of the chromosome. Steps for VNTR analysis. First, the blood samples are collected and DNA is isolated from all the samples. PCR is done to amplify the DNA samples. This is followed by cutting the DNA at restriction sites. Care should be taken that the restriction site used is on either side of the VNTRs and not within the VNTR. The fragments created with the restriction enzyme would be of different lengths for different individuals depending upon the tandem repeat length. This is followed by separation of the bands by gel electrophoresis. Then Southern blotting is done and the pattern is visualized using specific probes and compared. The figure here shows the VNTR profile of three individuals using three different VNTR or mini satellite markers. One can notice the polymorphism seen in the three individuals using the mini satellite marker. Use of VNTRs to create linkage map. Consider the pedigree given here. The shaded individuals are affected or show characteristics. VNTR markers A, B, C, D, E, F, G, H, and I are used to analyze the DNA profile of individuals. Inferences that can be made from the profile. A and B are allelic form since in the progeny either of the one is inherited. F and H are always linked together. Hence, they are in the same linkage group. A and D are unlinked. The profile also shows that all the affected individuals show VNTR for I and C. Hence, it can be concluded that the gene assumed to be responsible for the disease is linked to the VNTR marker I and C. This is how a linkage map is created using VNTRs. SSR or microsatellites. These are tandem repeats of shorter length, usually dinucleotide or tetranucleotide, distributed throughout the genome. The advantage of these over VNTR is that it is uniformly distributed in a genome. The occurrence of VNTR sequence frequency is more towards the telomeric regions. Secondly, since the length of microsatellite markers is less compared to VNTR markers, PCR typing is easier and more accurate. SSR are used in the same way as VNTR markers for constructing linkage maps. SNPs, single nucleotide polymorphism. These are markers which are different by only a single nucleotide sequence. Example, in the figure given below, nucleotide G is replaced by C in allele 2. The advantage of this marker is that it is uniformly distributed throughout the genome. Some can be used to construct RFLP markers. SSR markers are analyzed the same way as SSLPs for linkage studies to construct linkage map. The figure here illustrates the difference between the DNA markers studied. Single nucleotide polymorphism is observed in allele 1 and 2. The base pair CG is replaced by AT. This leads to variation in tandem repeats in SSR and VNTR profile. In RFLP, due to this, a nucleotide change and extra restriction endonuclease site is created in allele 2. Thank you.